So in this exercise 4, Meroplankton, factors affecting fish embryogenesis. So in this case, our group choose the title, How Different Percentage of Vinegar Will Affect the Development of the Fish Embryogenesis. So what is meroplankton? Meroplankton is an organism in which they spend only some part of their life as a plankton, and they will metamorphosis and became the next stage, which is the other form of the organism. For example, in this case, the fish at the first place it will become the plankton, and later it will become a fry, which is after the metamorphosis. So we use this fish. Anabastasides under the family Anabantidae. So the common name for this fish is ikan puyuh or climbing perch. So this fish is often found in swamp, lakes, canal, rice paddies, and stream. So this is all the information that have been gathered during the ingestion of a prim to the fish. So the fish will be transferred into this tank for them to spawn, and as we can see here, there is the tiny clear dot is the eggs of the fish after the spawning and after they fertilize. So the clear one is the one that we will take, and the black one that is denser, we don't take them as they are some sort of not in the good form. So the two main objectives is to study the embryogenesis process of planktonic larvae fish egg, and to study how different percentage of vinegar will affect the development of the fish egg.
So now we proceed to the methodology part. Okay, this is the flowcharts of the preparation before the experiment, where the 3 beaker was labeled with control 0.01% and 0.001%. Beaker 1 with control was added with 200 ml of dechlorinated water, and the parameter are first produced by the serial dilution. So this is the video of the preparation of the solution, where 10 ml of vinegar was measured by the using measuring cylinder. Then was put in the first beaker, and 190 ml of decorated water was filled in the beaker. Then the water was mixed with the dropper. The step was repeated until lesson 2, 3, 4, and 5. So this is the step during experiment where the method of crosstalk of Embryogenesis was injected with the overprim, the hormone which can induce spawning. So after injected, the broodstock was put in an aquarium in a dark place. So this is a picture of the injected hormone. So tank B, this is the information, and there are 2 male fish and 1 female in the tank B, and the tank D where they 2 male and 1 female. Why the 2 male? Because the size is more than female.
So this is the results of the embryogenesis lab. So you can see on the screen right now, this is the 2 cell formation, which can be seen happening at the same time for both control and treatment 1. But for treatment 2, it is not available due to the development of the cell is a bit fast than other. So this is the 4 cell development for both control and treatment 1. This is the 8 cell formation, which can be seen for control and treatment 2. For treatment 1, the image is not available due to human error happening during this moment. For treatment 2, it can be seen that 8 cell already it only took 0.58 HPF to achieve during this time. So this is the 16 cell development, which can be seen that almost all cells are developing quite at the same time. And so this is for the 32 cell, which can be seen that treatment 1 have a longer time than other treatment. So this is the 64 cell development, which can be seen right now. Treatment 2 is taking the longest time to achieve during this development process. This is 128 cell development, which can be seen that the most fastest treatment goes to control, which only took 188 HPF, 1.888 HPF. So this is the 256 cell development, which can be seen that almost all have an average time. For 1K cell, treatment 2 took the longest time to achieve, while control and treatment 1 have quite the same time. So for oblong, all have quite an average time during development of this process. So for sphere, it's the same goes where almost all control treatment 1 and treatment 2 have the same time. So for the dome process, treatment 2 took quite a time, but still on an average scale for other cell. For epiboly, control took quite a long time to achieve this stage, but treatment 2 is already quite having quite fast time to receive to be on this stage. But treatment 1, there are no image available due to human error. For 55% epiboly, we could see that treatment 1 and treatment 2 have quite an average time, but control is taking quite a time to achieve to this stage. For germ ring, we can see that all have the same time, but control is having quite a fast phase change from before. For shield, treatment 2 took quite a time to go to this stage, while control and treatment 1 have the same time. For 75% epiboly, we can see that treatment 1 has the longest time for HPF to go to this stage, while control and treatment 2 have quite an average time. For 90% epiboly, we can see that treatment 1 still have the longest time to go to next stage, while control and treatment 2 have quite the same time. For bud, we can see that treatment 1 is having the longest time to achieve, while treatment 2 is second, and the fastest one is control. For trisomite, treatment 1 is still taking the longest time to go to this stage, followed by treatment 2, and the most fastest is control. For tensomite, it's the same as before. Control is the most fastest one to go to this stage, while treatment 1 is the longest to go to this stage. For 14 somite, it can be seen that the most fastest is control, but it is changing, where treatment 2 is the one who's been taking the longest time to go to this stage. For 20 somite, the control is still the most fastest, followed by treatment 1, but treatment 2, the experiment has been stopped because it goes from the 10 o'clock to the evening, where there are still no changes of embryogenic stage. So for 26 somite, we can see that control took the longest time, while it is a bit faster for treatment 1. And for prime 5 for hatching, the fastest goes to treatment 1, while it took quite a time for control for hatch. So this is graph stages of the embryogenesis stage, where it can be seen that the most fastest embryogenic stage go to vinegar 1.
So today I will talking about discussion about Meroplantonic laboratory.
So first, I will talking about the embryogenesis of fish egg. First, after fertilized, the fish eggs will undergo the zygote stage, which is the newly fertilization eggs through the completion of the first zygotic cell cycle. After the zygote stage, the fish egg will undergo the cleavage when the cell cycle occur rapidly and synchronously to form the layer and organ. After the cleavage stage, the fish egg will undergo the blastulation stage, which is this Anaba species. They will undergo the meroblastic cleavage, which is the cleavage at animal poles lead to blastoderm or caps of the cells. Blastoderm overgrow the yolk epibolite, eventually enclosing it to form. This means the meroblastic cleavage, not the like holoblastic cleavage. With the holoblastic cleavage, if the overall cell will develop, but meroblastic cleavage only the half will develop. After that, they will undergo the gastrula stage, which is a hollow sphere of cell containing yolk with a small opening in the perivitelline space, the blastophore. Morphogenic movement of the involution, convergence and extension from the epiblast, hypoblast and embryogenic axis through the end of epibolite. And the last stage is for this experiment, we undergo the segmentation when the somite, branchial archaic primordial and neuromers develop. Primary organogenesis, earliest movement and the tail appears. So for this experiment, our group was using vinegar as the parameter of treatments. We use vinegar because to study the effect abiotic parameter, which is pH of the vinegar on the fish egg. So do the vinegar give the side effect to the fish egg? Based on our observation during this experiment, fish egg or embryo in the control beaker and treatment 1 beaker, which had third dilution, was hatched successfully. Duration time of hatching of the fish egg between these two beakers also same, but fish egg in the beaker 5 dilutions having a problem to hatch, which take a long time to change from the phase to another phase, and the last phase for fish egg is 14 somite. So this is our vinegar that we use in this experiment, which is the ingredient is water, acetic acid and salt. So impact of the vinegar to the environment, as we all know, vinegar is stable compound on the Wilson risk scale with the zero of reactivity, only harmful to animal, plant, aquatic if released in high concentration, and relatively low impact to the environment. So most suitable parameter for this experiment, there are many parameters to consider when they're culturing Anabas statidus, and they are known to withstand harsh environmental environment based to the Second et al. 2015. Parameter chosen in this lab are pH, where according to Ali et al. 2019, the optimal pH are between 7 to 8, while according to Dedi et al. 2016, the optimal temperature is 6 to 7 pH. In this lab, the pH used are 5.57 and 6.89 pH during the beginning of the experiment. According to Zalina et al. 2011, pH will not affect largely on the stocking density unless there is an increasing in stock or number. From the result, it can be seen that treatment 1 have faster rate of hatching. This happened due to presence of acetic acid. Acetic acid are known as ingredients for acidifier that are added in the fish food to help the growth based on the Lockstead 2018.
So as for the conclusion, so all the treatments, including the control tank, have successfully achieved all the embryogenesis stages. So for the treatment 2, on the other hand, the treatment is only reached until 14 soma as the process have been stopped there. And overall, the pH do not alter much all the embryogenesis process, but if it is exposed or released into high concentration, it will disrupt the process of the embryogenesis. So this is all the list of references that we used to do this video. Thank you.